It's a real honor to be here, and I honestly I wasn't sure what to expect, but to see an audience like this, it's really humbling because there's so many people that I know have an interest in how the brain works. The ones that I'm used to seeing are in their 20s usually. There's maybe a couple of you like that here, but that shows me that the interest in neuroscience never stops. And in fact, there's been a groundswell of interest that I've seen in the classrooms for 10 years. It's been getting more and more of a focal point for our biology students. I've been teaching, of course, I thought about this on the way down here in the car. 30 years now to university students, I've been teaching about anatomy and neuroscience courses. And some of those students are nursing students or medical students, biology students also, and even physical education students have taken my classes. So a wide range of people have shown a great interest in how the body works, but especially how the brain works. So thank you, Bud. That was really a nice introduction, and I hope that you have some interesting questions to ask me. I do want to tell you that I'm not a clinician. My training is in science of neuroscience. So I study, for example, I'll show you, I study how the brain of a goldfish works. And we use goldfish as a very interesting model system for how the brain of a human who maybe has a disease process could work. Because I'm not going to go into great detail the evolution of the brain, but things that I've learned about show me that our brain is not that much different from all of the other animals that are so-called vertebrate animals. And we can see things. I see it in my lab all the time. Similarities between quite primitive brains, like a fish brain or an amphibian brain. And really what we've done over the years, I'll summarize it into one sentence, is that we've added more and more things to the primitive form of the brain, but you can still recognize that same structure that a fish used for their basic needs in our brains, but we have a lot of other things that have been added onto it. So that's an interesting process that's taken hundreds of millions of years to evolve. And it's taken, well, me personally, 30 years to sort of get comfortable with. So let me grab my notes here, and I'll come stand back here, and we'll start. I'll start showing you the components of this lecture. What I really want to tell you about is how over this 30-year period, to me, personally, I see changes in the whole approach we take to teaching neuroscience. And not just neuroscience, because most of my students that take it overall, I would say, would be health professional students, nursing, medical students, and dental students. So how we approach the teaching of this very complicated material has changed, fortunately, as our technology has changed over the years, but also how our emphasis has changed too. And I can tell you that when I first started this, like generations of neuroscientists before me, people used books. We had books in front of us, and some of those books were black and white books with pictures that were made 100 years ago. And we'd stare at the books, we'd learn the names, hundreds of little arrows pointing to the structures, and that was the main emphasis. What is the brain? What are the different parts? And we don't necessarily know about everything, that the function of all those different parts. But we do know that if you injure certain parts, you get problems, you get deficits, and those are the first things that people learned about. What parts of the brain are responsible for certain symptoms if you have a stroke, for example, or if you get a traumatic injury to your head because of something that hits you in the side and you injure a piece of your brain. So for years, that was the majority of what we focused on. So we still do that. This is an illustration that's kind of like what we would normally teach our students 10 years ago, 20 years ago. Okay, so this picture is from a textbook. It's not a bad textbook. It's actually pretty good because it's in color now. It's not black and white. But you can see definitely that learning neuroanatomy and other parts of neuroscience is a very visual endeavor. And we learn to recognize what are these different structures. In this particular picture, they highlight them to make it interesting to see and to make it easier to point out. But, for example, in this instance, we look at these structures which are here, which are called the basal ganglia. And I'm going to talk a little bit about the basal ganglia, but next week when we talk, I think it's next week, we do Parkinson's disease with Dr. Barker's lab. discussing that. The basal ganglia are extremely important in how we control our motor activities, movement. And when there's disease process in some of those structures, we have difficulty moving. And okay, so basal ganglia include a number of different programs. And as it says here, we learn and we execute motor programs in this region here. And I really love to tell my students that one of the first motor programs we learn occurred before we're even born. And that happens when we are in our fetal period. And most of you already know this, that when a baby is born, the first thing that it has to do, besides cry, is it has to have a drink. It needs to feed. So during the last stage, maybe the last 2 or 3 months of gestation, this part of the brain is developing too, and it's developing a motor program for drinking, for sucking, and for swallowing as an essential component of the first thing that we need to do to survive. So the basal ganglia are early in their development. This is a cross-section of the brain. You can see a cross right there. I'm going to ask you quiz questions maybe. If you ask me questions, I'll ask questions back. But this area here is a horizontal section through the brain, and we can see the things that you may notice around here is that the brain, this is the cerebrum of the brain. The cerebrum is the main part of the brain that humans have quite an enlarged structure. There's a cerebrum in a fish. I use it all the time in my studies, but it's very small. It doesn't have these funny little bumps and gyri and sulci, which are the grooves. In other words, we have a great deal of surface area, and there's millions, billions of cells packed into that cerebral cortex. So the brown region is the cortex. That's where the thinking takes place, where our memories are stored. And all of these cortical areas, they interact by sending axons down through white matter, which is this white or lighter colored structures, and they communicate through synapses, chemical connections, neurotransmitter connections that interact with things like the basal ganglia. This area here is an area that's called the thalamus, and this is an area that communicates back from the basal ganglia through the thalamus and sends feedback information through different parts of this up here, actually, the motor areas of the cerebrum that control our individual movements. So that when we learn a motor program, like walking or jumping or swallowing or talking or anything that we do that we have to practice and learn from an early age, the basal ganglia develop a motor programming which is beyond our actually understanding at this point still. We don't totally understand what this word motor program means. In my mind, I believe it means that there is a network of axons and connections with synapses between this area here, for example, the thalamus, and the motor areas of the cerebral cortex, and back to the basal ganglia in a circuit. And there's different parts of that circuit devoted towards this kind of movement. And there's a circuit for talking, like I'm doing, and all of the different programs we learn to do. Okay. So, like it says here, when we damage this, we can get certain disease situations. Parkinson's disease is a well-known and well-studied pathology of the basal ganglia, and there's a part that's not even shown on this picture that is really functionally tied into those purple and red structures. It's called the substantia nigra. Many of you probably have heard that word because we all know examples of people who have Parkinson's, and I think now it's becoming almost common language. Substantia nigra means dark substance, and that contains a certain type of neuron called the dopamine neuron. And Dr. Barker he studies what happens when dopaminergic neurons die in the disease of Parkinson's. Okay, so there's also other diseases like Huntington's chorea, which is a very debilitating and deadly disease, also has problems with movement, but quite different than Parkinson's because slightly different parts of the basal ganglia are injured. In Huntington's, they give quite a different response. Okay, let me just go to the next slide, I think, because I don't want to spend too much time talking about Dr. Barker's research. I'm going to show you a video, and I'm supposed to stand here so that I get recorded, but I have to go back and forth. Okay, what am I going to show you? There's different parts of the brain that are really, really hot topics in neuroscience research, especially with disease conditions. And they're listed, some of the most important ones here. The cerebral cortex I pointed out to you, it's that darker area around the outside rim of the cerebrum. This is where the thinking takes place, our planning, our emotional personality traits are there, and our decision making, very important there. When somebody has an injury to the cerebral cortex, like a stroke, there's all kinds of very interesting disease symptoms that arise, and it's a very difficult area to repair. The brain is resistant to repair in so many ways. We know that now that the reason that repair is so hard in the brain compared to other parts of, let's say, the nerves that go down your arm or your leg is because the neurons in the brain don't have a protective covering quite the same in the brain compared to the peripheral nervous system, which can be repaired. But the cerebral cortex is subject to lots of injuries. Stroke is the biggest one. Traumatic head injury is another. So I can't remember the name of the researcher that's talking about post-stroke injury. Dr. Miller, and that's later on in this series. We'll be talking about this. This structure here is called the hippocampus because of its shape. Reminds people, the old anatomists, of an animal that looks like a horse or a seahorse, actually, the hippocampus. And the hippocampus is very important for learning and memory, and people who have Alzheimer's disease have early damage to this structure. And that means that their ability to form new memories and maintain memories is compromised. So that's a very intense area of research. What about the hippocampus, and why is it so susceptible to this early damage? Okay, this structure here, nucleus accumbens, is you probably have never heard of that, the word nucleus accumbens. One of my professors was an expert. We called him Dr. Accumbens. It's the reward center of the brain. And so it's a part of the emotional control areas. When people have addictions, they have pathology in their nucleus accumbens. And so they have overactive, kind of overactivity in certain ways with their electrical activation of their nucleus accumbens. They have a tendency to be overreactive, and they seek reward, like drug addiction reward or alcohol or gambling or any kind of addiction kind of thing. But it's also present in the general population that is normal behavior, because we all have desires to do things in a proper way, or we want to succeed at work. So the nucleus accumbens is at work all the time for directing us towards our most necessary and proper activities. Okay, and finally, I already mentioned basal ganglia there for Parkinson's. We're going to go on from that. Okay, so this is what that picture up there that I showed you looks very similar to what I learned with all of those structures. And now I'm going to show you something new that I've done, which I developed, and Bud mentioned that I had a Curricular Innovation Award with some money to do to create a special learning environment. And the main focus of this environment at first for me was for my science students, my clinical neuroscience science class. And the problem is with neuroscience, unless you're in medical school, you can't get access to a human brain to do dissection and handle the brain. You're not allowed. The coroner of BC will not allow students that are not medical students to go in. There are some other students that are affiliated with health professions that get permission to go in, but my students cannot. So coming from a medical school, before I came here, I was at Dalhousie Medical School. I did brain dissections with from human cadavers all the time. And it was the most beneficial and, I would say, memorable parts of learning neuroscience for my students back then. And I wanted my students in science at UBC Okanagan to get that benefit. So I asked them permission, and I got them to give me permission to take videos of me doing the same dissection I did for the medical students and the dental students, and me talking over the video program. And it was fun to make. We made, 2 hours of video programming, and we boiled it down to 35 minutes, 6 videos. And I'm going to show you one video, one of my favorite videos, in a moment. So there's me, obviously. Where I'm standing is in the Reichwald Center, the Southern Medical Program building on campus. And it's in the gross anatomy lab. And that's where the students go in to do their dissections of the human body. These are donated bodies, and they remove the brains from the skull. And they give me access to a couple of these brains, or 3 of these brains, and a big knife. And I had a schedule, and I said, okay, I'm going to show the following 60 or 70 structures. There's a video where I look at the surface features of the brain after we first remove it from the skull. It's not as exciting, but it's very informative of the coverings of the brain and how fluid circulates around the brain underneath the skull and then finally gets returned to the heart. And then there's a video on another surface views of the cerebrum with looking at the blood vessels that supply arterial blood to the brain and venous drainage back, that kind of thing. And then there's one on cranial nerves. All students have to learn the 12 pairs of cranial nerves and the spinal cord, all the different components of the spinal cord is very beneficial. And I like this one, mid-sagittal view of the brain. I'm going to show you this one. And if I was Dr. Barker, I would be showing you the horizontal sections because that's where the basal ganglia are with the Parkinson's connection. Now we use, a modern class like I'm hoping I'm developing with interactive tools. So part of what I've developed is these videos to give students a chance to look at and see what it's like to have a real brain, a human brain, and dissecting and see the different parts. And then we're going to go here to Interactive Neuroscience site. And I'll show you what I've developed if I can find it. Notice. Interactive Neuro Lab. This is another tool that we use in the class to help students learn the complicated names and the different structures that they have to see. Now this is, unfortunately, it's also presented so that I can't see everything, but that's okay. So this is a side view of the brain. And for those of us who are interested in the cerebral cortex that is around the surface of the cerebrum, all of these different places have special meaning. Like, for example, that's a part of the brain, it's a lobe called the parietal lobe. There's another lobe that's up here called the frontal lobe and the temporal lobe and the occipital lobe. And we learn all of the different major functions that are in those different lobes. And the way students can learn this is they click. There's a list here of different structures that they're supposed to see, like, for example, the lateral fissure. Now, we've tested this twice now. The first couple of years, actually all of the years that I teach neuroscience, I give quizzes on neuroanatomy. And I show them pictures, and I ask them, what's the name of this? What does it do? What does it connect to? Those kind of things. And they have a hard time. But since I've activated this program, they have incredibly high scores. They just really like doing it, and they get maybe 94% on the average. So it's been a very successful way of integrating technology into what used to be a very static, kind of boring way to learn. Okay, I have highlighted here some of the diseases that are important. I use these diseases in my course, Clinical Neuroscience. We study the nature of the disease, what's damaged, what the causes of the disease are, what possible treatments are available, what's the mechanism of the disease. Is it because of something in the cell biology of the cell itself, the neuron, or is it something different altogether? This is the most challenging part of any of the neuroscience courses, to figure out what's the cause of the diseases. I study epilepsy in the course, which is, of course, seizures, the chronic uncontrollable seizures. Autism, which I have highlighted in red because I know that there's a speaker coming. Actually, I highlighted it because nobody else is going to be talking about it. So I thought, okay, I'll give you some examples of what we know about autism. And everybody is interested, I think, in autism nowadays. And of course, Parkinson's, Alzheimer's, etc. So let's look at some things which I'm teaching my students right now, actually, about autism. Now, if you're not familiar with autism, it's not hard to hear about it and to read about it. Children who have autism, they often develop poor developmental landmarks, milestones, sometimes as early as the first year, quite often, more often, I would say, between the ages of 2, 3, and 4 years old. They don't develop the skills that other children would have. They have a hard time learning language. Or if they had language, they stop speaking. They regress. And oftentimes their social the ability to socialize is compromised. They don't communicate well, either verbally, of course, or in other ways. Even sometimes looking at people is difficult for them. I think most of us have somebody that we know that has autism spectrum disorder. It's a complicated mixture of multiple causes, and now it turns out multiple genetic mutations that are involved in creating the disease in the first place. I found this part particularly fascinating and relevant. This is a picture of the brain from the sides, similar to what I showed before. This is called the temporal lobe, and there's the frontal lobe and the occipital lobe back here. And what they've done in this picture, they've tilted the brain so you're looking at the undersurface, the ventral surface. And by the way, this part in green is called the parahippocampal gyrus, because that structure, the hippocampus, that's involved in Alzheimer's disease and problems with memory formation, lie in the inside of that hippocampal, that bump. The gyrus is a bump. Each of these grooves is a sulcus, each bump is a gyrus. So within that green stripe there of the parahippocampal region, there's a place called the parahippocampal place neurons are located. And when we walk around a room like this, if you stayed in this room like I do teaching, and you get familiar with all of the seats and the students, people were very smart. They figured out how to put electrodes into animals that have been trained to be in a room like this. And when they walk around the room, when they get to a certain place, one set of neurons will be activated, and then they move over to a different place, and right next to it there'll be another set. It's like a logical projection mapped out in the parahippocampal place region. It's where we develop our maps for the environments that we learn and are familiar with, the place area. This is not such a big problem with autism spectrum disorder children, but this area, which is in the fusiform gyrus, just a little bit above it, is the area where people, when they see faces or they show a picture of a face, the neurons in that area, they can do this with humans, and they use new technology called PET scans, positron emission tomography, where the areas that are most active light up with the computerized colored scheme. And normal people, when they're young, and as they grow, when they show them a picture of the face of your mother, your father, somebody they know, that area will become very activated. But with autistic children, these areas become not a nicely packaged zone like that. It's poorly activated and not regionally compacted. It's diffuse around, including that area, but outside the borders of the normal fusiform face area. This is characteristic of what I'm going to show you in a couple slides coming up. There's a problem with autism, they believe. It's now being discussed as the central problem for most children with autism is a difficulty in maturation of the synaptic connections. Now, I never knew this until I started teaching this course, but when we develop as a youngster, in the first couple of years of our life, we actually overproduce the number of neurons we need. We produce millions of neurons every minute, and they grow and they migrate to the proper locations. Some of them migrate to this zone, and then other areas send their axons, that are the communicating bundles, into those zones, and they form synapses. That's the communication network. And the synapses are totally important, but they're also overloaded. We start off in life with way more synapses than we end up in. So what happens during that developmental period? There's mechanisms that we're just starting to understand where those synapses are actually removed. The unnecessary, unbeneficial synapses are removed, and the neurons that give rise to them are removed. So the fact that an autistic child has a fusiform face area that's poorly formed is because there was an overgrowth of synapses that didn't mature properly. And the child has a problem when they see the face because it's called the signal-to-noise ratio. It's kind of something that we talk about when we see a picture. The signal is the picture, and everything around it is all the noise. And this area is diffusely organized and it's getting other signals mixed in with it that are not supposed to be there. And the child is confused and frustrated and tends to look away because they don't want to see people's face because they get a confusing message. That's my understanding of it. And this is I just read a wonderful paper about that. It summarized this very thing, and I'm going to show you some of the things that are going on. Now we're going to look at this, and don't get scared by this. This is a cell biology of a synapse. This is what people are investigating at the cellular level and the genetic level at the same time. Now what this is this is a picture of an axon. This is the terminal bouton, they called it, of a little swelling of an axon, and this is the receiving part, the dendrite. The dendrite communicates with the axon terminal and it forms a synapse. There's the synaptic cleft right between them. And you can't see it in this picture. I wish I had it in there. I should draw it in. There's neurotransmitter packages here. And normally what happens is that this axon releases that neurotransmitter and it binds to receptors down here which create a response. But the problem is that we developed way too many synapses at first, and then between 2 and actually puberty, 2 years old and puberty, the number of the extra synapses slowly gets taken back. They get pruned. That's the term they use, like pruning the apples off a tree. When they sell apples around this town, they prune their apples so that only the best apples are left, and the ones that they don't want, they get rid of. Same thing has to happen here. The things that hold the synapse together, that makes it difficult to prune, are these proteins that are holding the presynaptic axon terminal to the postsynaptic dendrite. You can see these proteins, they have funny names, but they make sense. This is called synaptosomal cell adhesion molecule, SYNCAM. It's embedded in the membrane here of the postsynaptic dendrite, and it attaches to another one up here. So it's like a handshake. They call it the handshake. And that handshake stabilizes the synapse and holds it together. And during the early stages, there's lots of that, and they're very stable. If they're too stable, they cannot be broken and they can't be removed. And the synaptic connection, doesn't disappear. Here's another one, but these are two proteins here, neurexin and neuroligin. And I'm pointing out the names only because they've found that autistic children are associated or linked genetically to mutations in the gene that makes neuroligin or neurexin or SYNCAM. Those are 3 proteins that have been associated with a genetic mutation that is a high risk for autism. And here's another one. This came out just a little while ago. This protein here called CASC, and I forget I don't even care what the name of CASC is. K means that it's a kinase which adds a phosphate group to itself right there. And this is a tightly regulated process. This enzyme here causes this CASC protein to slip away from its little dock that's waiting. The CASC diffuses down and it binds to and it pulls the SYNCAM and the neuroligins and the neurexin together, and it stabilizes the synapse because CASC is the organizing principle, the protein that organizes those little proteins like glue to come together. CASC and these other cell adhesion molecules, genetics has shown that there's a higher incidence of mutation in those than other proteins. And even this CASC is associated with this thing is called an ion channel for calcium. The whole function of the axon terminal is dependent upon the localization of this calcium channel, allowing calcium to come in when an action potential electrically activates this axon. And the calcium is responsible for causing neurotransmitters to drift across and create the response down here. So CASC, I would say, is a fundamentally important protein for the function of a synapse, and it's also for the establishing the synaptic connection and holding it in together. If we don't have the proper balance between creation of the synapse and then pruning or removal of the synapse, then we get into difficulty. Okay, this is a picture that shows something else. This is the postsynaptic part. This is the dendritic piece. It's called a spine. This is where the synapse takes place, right on this dendritic spine. And each of these little brown dots here is a colorized, stained image of one of those little spines. The more of those little dots there are, the more synapses there are. This group here, which group is it? I have it down. It's probably the next group. It's Tang et al. It's a Southern California group. They found that there's another protein that's been mutated in many of the autism spectrum disorder children, and there's SHANK3. It's a funny name, but this protein, it holds together a different group of proteins. These are the actual neurotransmitter receptors, in this case for glutamate, which is an excitatory neurotransmitter. Without SHANK, there will be no synaptic response at the end. So there's always got to be SHANK there. Well, they found out that there's mutations in SHANK3 in almost 1% of all the autistic children they've found. So there's a problem with establishing this neurotransmitter postsynaptic function there. So just one more example of blending genetics with cell biology and neuropathology, which is the autism problems. So let's look at the next slide. I'll show you an example of what happens. The cortex of an ASD child has too many synapses. Okay, too many synapses. So, all right, so 7 o'clock. So I got to move on here. Here's an example of, this is the number of synapses. The higher on this y-axis, this is a dot for each synaptic spine that's present. And you can see the synaptic spines there. In a normal child, that's C for control. These are post-mortem brain studies of children who have died for a reason that's not associated with their disease. They were in a car accident or they had a heart disease or something. So when they die, they have autism. They get permission to take their brain and autopsy the tissue and look at the cellular structure, and they look at the spine density. Normal child, it's high in the very young. This is years old, 6 or 8. And then after 13 to 19 years old, it has gone down by like 50%. This is the maturation process that's normally happening. Now here, A is for autism children. There's 10 of them that have been studied. They start off, once again, actually a little higher than the normal child. And then over the 13 to 19-year period, there's hardly any pruning or removal of the synaptic spines there. So this is an indication of synaptic pruning may be fundamental to the kind of symptoms that children show where they have too much activity. They have confusing hyperactivity of their cerebral cortex in some parts of their brain. The parts of their brain that are associated with language communication and comprehension, and some other interesting things. So let's look at this area here. That study was done in this part of the temporal lobe because the auditory cortex, where you understand sounds, is there. And this part right here is the part of the brain that's where you have oral comprehension of languages there. That's where they found this problem with the synaptic pruning to be the greatest, here and here. So overformation early, pruning is absolutely necessary for normal development. If you don't get that, then you get symptoms that are associated with confusing, conflicting synaptic communication going on. Okay. Let's look at something else. Here's another thing. I'm going to go through this. I'm going to skip this one, I think. This shows you, once again, a different color of the different dendritic spines. But there's a protein here called mTOR, which I know what it stands for, but it means nothing personally to me. But they know that it inhibits the part of the cell that is actually removing those synaptic spines. That's the maturation thing, where spines get taken away, the synapses, the extra ones, are taken off the map. mTOR is responsible for that process. Actually, mTOR inhibits that process, and in Alzheimer's autism children, there's a hyperactivity of mTOR. There's too much. So the process that's normally supposed to remove those extra synapses is non-functional. So this is a really great study. It shows that that this is a possible mechanism maybe for future treatment, because there's a drug that they've established that they can block the activity of mTOR and restore the number of spine density that you're supposed to have. And when they test this in an animal model, the animal at first is like a autism behaving animal, and they give this drug, and it simply, all the spines disappear to their normal level, and they go into the normal behavior routine. So there's a lot of really good potential mechanisms for drug treatment in the future once they understand this. Okay, I have until 8:30, and we lost 5 minutes because of my video thing, but this is a good time to ask if there's any questions about what I've said so far, and then I'll go on to look at my research with the fish. So don't feel shy. If you had a question about what I've talked about, feel free. Just remember, I'm not a clinician. So I don't have all of the answers, but I have maybe some. Okay. You can think about it. We'll come back at the end, and you can still ask. Okay, let's look at what the Mathieson lab is doing. We're looking at goldfish because I told you the goldfish shares a lot of the neuroanatomy and the neurophysiology of parts of the brain, just like we have. Okay, so what I'm looking for is I want to know what controls the growth of new neurons in the brain during embryonic period, but also through our life as an adult. Our brains, we don't grow our brains as an adult very much, but some of the old neurons get removed because they're damaged, and we have a mechanism in place called neurogenesis for replacing the old damaged neurons with new functional neurons. So neurogenesis is what I'm looking for. I want to find out what controls growth and repair, because let's face it, a lot of people get injuries to the brain, and even more so to the spinal cord, and the same thing happens in the spinal cord. We need to figure out how to control the birth of new neurons in a damaged part of the nervous system. Okay, so this is way beyond what I'm going to tell you about, but this is about a type of hormone called steroid hormones, and we all know what they are. They're normally associated in our thinking and our speech with the gonads, testicular steroids or ovarian steroids like estrogens and testosterone. But some of you would be interested to know that the brain is not only a target for steroid hormones, and it's actually responsible for some of the differences in our behaviors between men and women because we're exposed to different steroid hormones early in our life, and it changes the synaptic maturation in a man's brain versus a female's brain in ways which create our different behaviors, which people argue about all the time, right? They're still trying to decide if we're from Venus or from Mars, but it essentially comes down to steroid hormones in the early part of our development. Well, plus the things you learned from your mother and father too, I suppose. Okay, so steroids, they can enter the brain from the blood and cause a change in the development of the brain, but they can also be made in the neurons themselves. There's a class of steroids called neurosteroids, and neurons have the enzymes in them to manufacture those steroids. They make them from a molecule which we all love and hate called cholesterol. And cholesterol, there it is there, goes through this long sequential change to molecules like pregnenolone and then progesterone. This is how progesterone is made in women, for example. But in the brain, the same thing happens, and progesterone gets converted by this enzyme into dihydroprogesterone, and then another enzyme into this one, which is what I study in my lab, allopregnanolone. This is like the poster child of neurosteroids in current research. People have found that neurosteroids influence the development of the brain, spinal cord, the physiology of the brain because of the electrical connections that it makes. It's important for epilepsy and controlling epileptic activity. It's important for now being used for treatment for diseases like depression and schizophrenia. It's amazing. It's the wonder drug of neurosteroids. Okay, I love this drug because it's also important for the growth and the repair and connections between adjacent neurons through their axons and synapses. Okay, so there's neurogenesis. That's the process of making new cells. And how do we make that? We make that even in a fish. There's populations of cells that are just like an embryonic cell. They're called stem cells, neural stem cells. And neural stem cells, they can't make blood or they can't make muscle, but they can make neurons and the supporting cells called glial cells. And what controls that? We think neurogenesis is controlled by neurosteroids. There's evidence that came up to show that neurosteroids are very important. For example, well, we know that in the adult, there's only a few places in the brains of a human that have neural stem cells. Most of them disappear. And in humans, we have the hippocampus, the learning and memory part, and oddly enough, a part of the brain that creates new neurons for the olfactory senses. Well, people tell me it's because in our olfactory system, we have to replenish old neurons at a very rapid rate. We constantly have to replace olfactory sensory neurons, so we have to replace the neurons that connect with them too. And that makes sense. And the hippocampus, they say, we need to create new memories every day. So we have to make new neurons to allow us to make new memories. And the hippocampus is where we start the process of making memories. So the stem cells, they keep dividing, and then they are tweaked, and they go off and they start changing from stem cells into what are called neuroblasts. They're primitive neurons, and then they differentiate and mature into all the different 500 different types of neurons that we have in the brain. Okay. In humans, like I said, there's only 2 of them. That means that, unfortunately, if we injure the brain, it's really hard to repair injury to the brain because there's only a few places where stem cells are located. In the fish, however, there are for reasons which are debated, there are dozens of places that have stem cells. And I see them in my lab. I cut them and I stain for them. They're all over the place. Okay, neurogenesis is very strong in the fish brain. So I'm using this as a model system for figuring out what controls neurogenesis. This is the cerebellum of the fish brain cut in cross-section. And like I said, the fish brain is beautiful and the cerebellum is great. This is a dark blue stained. There's millions of neurons packed in there, and there's lots of white matter and axons up here. And there's stem cells right there and in this zone here. This is 2 different parts of the cerebellum. And at the highest place in the fish, there's more neural stem cells there than anywhere that I've been able to find. And we can actually measure that. And we measure it by looking at cell division. And this is not the brain. This is actually a piece of a fish intestine. But we know that intestines always get new cells, even your intestines. There's always old cells being removed and new cells replacing them. So we have to test this with an intestine. But we need a special dye to do that. So what we do is we incubate by injecting this drug called bromodioxyuridine into the body cavity of the fish. It goes into the blood, and it crosses the blood-brain barrier, which perfuses the brain of the fish. And every cell that starts going through cell division incorporates BRDU into its newly made DNA. When a cell divides, there's a new copy of the DNA always made. So now we have a cell that's divided within the last 2 hours, because that's how long I treat the fish for. And if I look for the BRDU molecule, I can see which cells are dividing. And you can see here that the BRDU has been tagged with a green fluorescent molecule and looked at in the confocal microscope, which shines a laser beam across the tissue. And every time the laser beam, which has ultraviolet light, hits that cell, it's got BRDU in it, and it lets off a little flash of green light. And we can photograph it and count those cells. So this is my control tissue, because I know tissues, this is intestine, always has cells that are dividing, and there they are. Now this is the cerebellum. This is that part of the top of the cerebellum. It's called the corpus cerebellum. It receives sensory information from the skin and from the eyes, actually. It's an area of active sensory input. And it's a depot of high neurogenic activity. Okay, so what they think happens is why the fish has so much neurogenesis taking place here and other places is because fish, you may know, are constantly growing. Their eyes get bigger as they grow. Their skin gets bigger as they grow. Their muscles get bigger. Every new body segment that grows has to be innervated by new cells. So a fish, as long as it's continuing to grow, has a high degree of neurogenesis. It's a perfect way to study, a mechanism to study this process. Okay, so here's another part. That's the lower box of that cerebellum called the lobus caudalis. you can actually see there's the zone where the stem cells are located here, and this is my interpretation of seeing this. They migrate quickly as soon as they're made. These are all labeled. Each one of those green dots is a nucleus of a cell that's going to make a neuron as it develops. And they migrate up and they move to the different regions of the cerebellum, and they set up shop, and they establish their synaptic connections, and they become functional. So that's what the benefit of this BRDU and the measurement of neurogenesis is. It's looking for new neurons. This, by the way, is the cerebrum of the fish right here. They don't think very much. They have a cerebrum like you and me, but they make some decisions. They make decisions on whether or not something they put in their mouth is good to swallow, like if it tastes good. They make decisions on whether another fish is going to attack them, or they have to swim away or fight back. Those are the kind of basic decisions a fish makes. This part of the zone of the cerebrum is just like your brain right now. You are getting new neurons develop in your olfactory area, and part of the cerebral hemisphere of your brain has neurogenesis taking place, and that's what that region is associated with. It's similar to olfactory neurons, and they're migrating down this olfactory tract, and they're going to establish their connections with the olfactory area in the fish under their little nostrils. Isn't that cool? Cool. I love that picture. It looks like an elephant to me, but it's just really great. I like it because there's so many cells that are dividing, and it's just like in the vertebrate human brain, even though we're very advanced. Okay, we see this in the cerebrum as like this picture. We see it in the optic. This is the region of the brain of the fish that the eye sends its information to, optic lobe, and down in the spinal cord too, although I've had a hard time finding it for some reason down there. All through the central nervous system, there are regions of new neurons being born. Now, why is it important? Well, it's because in some disease conditions, like in Alzheimer's disease, you're going to learn that the cerebral cortex cells die by the millions all the time. As we get older, that happens anyway, but in Alzheimer's disease, it happens at an accelerated rate. And here's a picture. It's a famous PET scan. I see it in books all the time. This is showing just the size of the cerebral hemisphere on a normal patient on this side with the gyri and sulci at an age maybe in their 70s. Post-mortem. No, actually, this is not post-mortem. This is a PET scan, I think, of a normal, living, breathing person. And here's a person with Alzheimer's disease, and you can see how shrunken down it's become. So it's lost a high percentage of its neurons, her or his neurons. Well, how can we get that to stop? They found that in Alzheimer's patients, when they do post-mortem tests, they find that, first of all, one of those neurosteroids called allopregnanolone that I had circled is almost depleted in a person with Alzheimer's. It gets low in all of us as we age, but in Alzheimer's disease, allopregnanolone declines and it's depleted in other neurodegenerative diseases too. It seems to be essential for maintaining the proper level of new neurons to be born to replace the dying neurons. So people are looking at allopregnanolone as a potential therapeutic mechanism for Alzheimer's. If we could figure out how to deliver it or at what stage of the disease to deliver it, we could bump up the number of new neurons, perhaps, and counteract the disease process which is killing them. That's how people like me are thinking. Okay, so can we treat Alzheimer's patients with allo? Not yet. Allo is just, in fact, the whole neurosteroid area wasn't even understood, never discovered until 1980-something. It's a new field, but there's a lot of activity in it. Okay, so one of the questions that is going on in my lab is we want to know, why do fish have so much neurogenesis? Is it because they have more allopregnanolone than a human being would have, for example, or other mammalian animals? And that's one question that's in red because that's what I'm looking at right now. We do that by grinding up the fish brains, and we measure how much allopregnanolone is in each different part of the brain. So today we're doing that in the lab too with these terrible chemicals in the chemistry lab, which I hate, but my student is learning. He's a biochemistry student. He loves it. So that's what we're going to answer. How much allopregnanolone does a normal fish brain have? Can we trigger neurogenesis by increasing allopregnanolone? So that's an interesting thing. If we can do that, would allopregnanolone treatment change the number of BRDU, those green neuron flashes, compared to a normal untreated animal? Or if you injured the brain, would allopregnanolone help the repair mechanism? So experiments that I do typically is we take an adult goldfish. That's where all those pictures I showed you were from an adult. We inject allopregnanolone a little tiny bit along with BRDU, that fluorescent molecule, and we let it sit for just a short time. We anesthetize the fish and sacrifice them and then remove the brain for microscopy. And we count how many BRDU neurons are there, and we can find out if allopregnanolone has effect. And this is results that my student found in the summer, Justin King. And this is a control fish with no treatment. And this is the number of, it's been standardized, this is the number of neurogenesis neurons that we count in a normal control fish. Allopregnanolone for 8 hours with this 1.5 milligrams per kilogram of fish, it almost doubles. Actually, is 68% or something greater than this one. So it's increased the amount of neurogenesis quite substantially. But we still don't know what the mechanism of allopregnanolone's actions are. So my biochemistry student, that's what we're trying to figure out. There's got to be a mechanism. Okay, so when we look at the possible mechanism, we look towards what controls cell division in the first place. This is a picture that many people who've taken biology see. This is the cell cycle with mitosis up here, M, and these are different stages. There's S is for DNA synthesis, and there's 2 gap phases where they prepare the cell for synthesis and prepare it for mitosis. Each of these stages, there's little blocks which stop them from going from one stage to the next. We think that allopregnanolone facilitates cell division by making it easier to go from G1 to S, and then from S to G2 or G2 to M. That's our hypothesis. So, it's a very important question because you can get too much cell division and you can get tumor formation, cancer, right? Uncontrolled cell division is cancer, well, in many cases. If you don't get enough cell division, an animal like a developing animal won't get the brain developing properly. It will be underdeveloped. So you have to get the proper amount of cell division. This is a very tightly controlled mechanism. And there are genes that are now being established that are controlling genes. They are essential for controlling the progression from one stage to the next. There's at least 5 or 6 that we have potential genes that we can look at. Okay. What controls those genes? Growth factors that are inside the cytoplasm of the cell or that are attached to the cell surface, and they create an intracellular signaling mechanism, turns on those genes that allow it to go from one division, a second division, a third division, etc. We're testing one of these things, which I'm almost afraid to put it up. It's called beta-catenin, but there's so many different molecules, but we know that beta-catenin is really a potentially excellent model. Okay, and we know that if we get too much beta-catenin in cells, tumor formation will happen. Not enough beta-catenin, the brain will not grow properly. And so that's what we're testing too. We want to know, is allopregnanolone acting in that graph I showed you by stimulating beta-catenin to allow mitosis to take place, or cell division. And here's beta-catenin here. This is what happens with, this is one of the best known, I'm supposed to be standing over here, sorry, Jeff. It's called Wnt. It's just a signaling molecule that's outside the cell. When it binds to this funny receptor called frizzled, and they have really strange names for the cell biology of any cell. When Wnt binds to frizzled, it allows beta-catenin to go into the nucleus here, and it binds to a piece of DNA there, and it activates one of those genes that controls the sequence from one part of the cell division to another. So these are transcription factors, they're called, that regulate the expression of gene activity. So beta-catenin, if there's a free accessibility of beta-catenin, a gene that's essential for cell division will be activated. Without beta-catenin, this is what happens. This is called the proteasome. It's the garburator of the cell. This is where proteins that you don't want to exist in the cytoplasm of the cell get funneled in, and they get chewed up and spit out as little tiny pieces. And if beta-catenin is allowed to do that, the cell will stop dividing. If beta-catenin is not allowed to go in there, it's free. It will bind to the DNA sequence and allow cell division to progress. So our hypothesis is, this is where I'm going to finish right now because I'm past what I was supposed to do. Our hypothesis is that we're going to look at treat allopregnanolone with our fish and see an increase in this type of beta-catenin, which is the one that's free and allowing gene expression of those cell cycle dependent genes. So thank you all for listening. I really appreciate it.